Can you hear me? Can you hear me now? Hello? All right. Um, yeah, apologies for the small technical difficulties here. So I've managed to join. I would like to thank you for joining here today. Um, I hope you've had a great start of the session. And my name is Yannick Djoumbou. I am a data scientist, a senior data scientist at Corteva in the US. And today I am going to provide a quick um, hands-on session. We're going to explore antimalarial drug through cheminformatics and QSAR. Because of the inability to send my instructions before the working session, what is going to happen is I'm going to do a live demo, a live session here where I'm going to go step by step through the different phases of this process, and I am going to be sharing the code via a link later. So I will provide the URL to the link to my the organizers, and those will be shared with you. That way you would have some examples of how to run this type of analysis. It's a very it's going to be a very basic set of tutorials, but then with additional links so that you can further explore the concepts that I'll present here. So, and let me know if you have any question. Feel free to interrupt. And if you can hear me for whatever reason, please feel free to let me know. Thank you. So, a brief presentation about me, a brief introduction about me. Sorry. So, I was born in Cameroon. This is where I studied for the first 16 years of my life. I earned my high school diploma there, particularly in Douala, for those who know Cameroon. And I went to Germany to study. So I obtained, my BSc and MSc in computational biology and computational chemistry. And then I traveled to Canada for my doctorate. So that's where I started exploring cheminformatics with application in biotechnology and metabolomics in particular. And after a short postdoc, I went to the US. So as you can see here, a few tools that I've developed along the way include classifier for structure-based classification, automated classification of chemicals. You might have heard of it. Biotransformer for metabolite prediction. So feel free to check those tools, and CFMID for the prediction of spectra, starting from compounds and also compound identification. You might have heard about the drug bank database, about the human metabolome database, and also the toxin and targets database. Those are projects that I've been part of while I was in Canada. Currently, I work for a company that specializes in agrochemical discovery. But here today, I'm going to be representing myself and not the company. So the views that I express here are mine and mine only. So I recently moved to the Bay Area in California. So I'm working remotely from there. So if you're interested, a few of my hobbies as Cameroonians, that's something that is not so, unheard of. So we are a lot into music and soccer and basketball and all that. So that's for me. And today we're going to, work towards the discovery of new antimalarial drug. Obviously, we're going to use an assay that relies on already tested compounds. But what we're going to do is, build a model to predict compounds that, have not been tested before. So you've heard about malaria earlier today, so I'm not going to say too much about it. As you know, it's a disease that is affecting a lot of people. For those who live in the tropical regions, the likelihood for you to have experienced the disease before is quite high. I've been a victim of it myself. As you know, it's transmitted between people via infection of infected mosquitoes. So the bite of an infected mosquito will then enter the victim's body and travel into the person's liver, and that's where it will reproduce, and the disease will progress unless stopped. So it can easily lead to death. So that's why it's such a critical condition to find cures for. So with respect to the drug discovery design, and in particular computer-aided framework, we could we could simplify that design as a heat, or that design process as a heat to active to lead process. So a heat usually is a compound that passes a certain activity threshold. For example, early binding, binding against a certain target, right? So an active is synthetically actionable. And he has activity against the target, but we can also quickly weed out or remove the ones that don't show an unwanted mode of action. So if the molecule is hitting a target of interest but also hitting a bunch of other targets and leading to toxicity, these type of molecules can be weed out early on. The lead usually has an activity profile that is significant enough. So it might be a novel molecule that has not been discovered before, or a molecule that is outside a certain protected IP chemical space, and something that warrants significant involvement. So, and depending on whether we have a priori information about the protein target or not, scientists will use several methods to maximize the likelihood of the discovery of new heat. So in best case scenarios, you will have the ligand structures, and you have the structure of the target, but also maybe ideas about known ligands, right? And then you go from there to design new ligands, or you will be able to dock a bunch of structures into the ligand of interest, and then select the ones that bind, and then from there you will further filter those molecules to prioritize them, maybe by physical chemical properties, or by additional activities, or by toxicity. And that you can use you can filter those molecules using predictive models, for example. And that's why it's important to have predictive models on top of things like docking stations, visual screening stations, right? So in crop protection, where I currently work in, the discovery in the earlier phase is mostly following a ligand-based design. So we don't even know the target. All we know is that, okay, we have a compound or a known ligand, either based on literature research or hypothesis. We have a ligand or a scaffold that is able to kill a pest of interest, or a pest that is genetically closer to the pest of interest. And starting from there, we will then build or find ligands or molecules that look similar in terms of shape, for example, or that have a similar profile in terms of certain specific properties. And that's where we start hitting the QSAR models, and then filtering those molecules away that are toxic or bad. And then we will pick the ones that are more interesting and go for further testing. So I am going to briefly illustrate the drug discovery pipeline here. It's similar to the agrochemical discovery pipeline. One thing that it's very important to notice is that the molecular discovery or small molecule discovery pipeline, whether it's pesticides or pharmaceuticals, they follow an iterative design, make, test, and analyze concept. So it's something that starts with designing, the first set of molecules. As I mentioned, it could be through docking or ritual screening or de novo design, and then they are synthesized in the make phase. They are tested in the test phase, and then they are analyzed in in this case, this is where, as you will see, QSAR modeling will appear or occur. So in the design, you can use the ligand structure or fragment based model, where, as I mentioned earlier, you design new ligand or you look for new molecules based on known actives, right? Or the structure based model could use the structure of a protein of interest, or information about the binding mode of known ligands of that protein, such as pharmacophore constraints and things like that, or binding pocket information to design molecules that would fit into the pocket. And obviously, the idea is to optimize the molecule so that it can reach multiple objectives. It has to, adhere to the right atomic properties. So it has to, be absorbed properly, distributed properly, metabolized properly, and excreted. But it also should not be toxic. And at the same time, it has to hit your target of interest. So now the make has to do with, finding. So this part has to do with finding the correct route that will lead to you building the tool, right? The compound, right? You create the compound, and there is a huge field now that aims at building software and predictive models that will take a molecule of interest and, starting from a library of building blocks that you have in store, for example, tell you what are the best routes, what are the best conditions to generate the molecule of interest. So then you test the molecule, right? So you test it against your target, against associated target, against targets that you don't want the molecule to eat, to hit. You would test it, for you measure some physical chemical properties like the solubility or the lipophilicity and so on and so forth. And then you capture the data. And then after capturing the data, you have to analyze it and generate further hypotheses to come back and maybe optimize the molecule through additional design. So in analysis, you have the QSAR analysis, the SAR analysis, or structure activity relationship analysis, and that's where you take advantage of the data to build models, so that you don't always have to go through these expensive tests, right? And then you also learn about the toxicology, and then you can also learn about the metabolism or environmental fate of your molecule, right? So with all of the data here, you can build models to then assist you in the discovery or the design of the better molecules, right? So, and as you can see here, it's an iterative process. And that's where QSAR is applied in the analysis phase. So what is QSAR? Basically, QSAR is a modeling technique, or a modeling framework that aims to establish mathematical relationships between structure properties of a molecule and its biological activity. So it usually provides a cost-effective and efficient way to estimate the estimate the activity. Now, generally, when you start with this drug discovery campaign, or small molecule discovery campaign, it could be a drug, it could be a crop protection agent. It could even be a molecule for cosmetics. When you create fragrances, sometimes those are artificially made, right? So you want to start with a huge number, and then find the most appropriate molecule. So you start with hundreds of thousands, if not millions, sometimes tens of millions. We're trying to go into billions of molecules that you start from, because the space of chemically feasible molecules of interest is, about 10 to the 60. So that's huge. So what you want to do, you cannot test all of them experimentally, even through high throughput screening. It's not going to be sufficient. It's going to take you, centuries, right, to be able to do that, and it's going to be very expensive. So QSAR models provide a cost-effective and an efficient way to estimate the activity of molecules. And it reduces the reliance on the expensive, time-consuming experiment, or delays it up to down to later stages. Now we have QSAR, but we also have QSPR. QSPR stands for quantitative structure property relationship. So this framework does not just allow to detect, for example, if a molecule is toxic against humans, or if a molecule binds to a certain protein. It can also allow you to calculate or estimate a certain property, like let's say lipophilicity, for example, based on a number of different descriptors, as we will see later. So QSAR modeling, as I mentioned earlier, helps researchers to prioritize the candidates for further investigation and development, and it focuses the research on the molecules that are most likely to be successful. So you want to start with the hundreds of thousands, if not millions and hundreds of millions of molecules, down to maybe a few thousands that you are going to focus more. So those thousands, you can then put them through high throughput screening, and then get the real data. And as you progress, the pipeline is going to be smaller and smaller and smaller, right? The pipeline is going to be smaller and smaller and smaller, and then you're gonna get down to maybe a few tens of molecules that you are going to push through clinical trials, for example. So the negative design here removes the undesirable compounds like earlier on, and then the positive design selects molecules with desired property, right? So the QSAR models take certain types of information and transform them or manipulate them to produce another type of information. So the molecules are generally represented in various forms. So you can represent it in SMILES notation. That's a one-line notation that is very useful. This is the most standard, the most popular representation, because it allows you to have an idea of what the molecule looks like. Of course, it's not as clear as the graph, but at least for the sake of ease of manipulation, and also even for natural language processing, the SMILES is very, very powerful. So we also have fingerprints. Right? Fingerprints provides an abstract representation of the functional groups or molecular features that are present within the molecule, right? So they are usually of fixed length. And they can be predefined. They can be based on structure. So you can have, for example, a list of 10, 20, 100, 1,000 different structures or structural fragments that you have identified as important to large-scale analysis. And you define each bit here for each of those structural keys. For example, the first bit could be indicating whether a carboxylic group is present in the molecule or not. The second bit will maybe indicate whether a primary amine group is present in the molecule or not. And you could decide to just identify the presence or absence with ones and zeros, or you can decide to count. So it could be 2 carboxylic group, 1 primary amine, and so on and so forth. So those are things that you can try when you are doing your predictive model. You're going through your predictive modeling project. So those are fixed, as I mentioned, but they are also learned features. They are learned from semi-supervised or unsupervised learning, right? So you probably heard of deep neural networks. There are other methods that are not necessarily deep neural, based on deep neural networks, that can allow you to take molecules and represent them into a set of features and provide a mathematical representation of the molecules. But those are not fixed. So depending on your data set, they can, the even if the vector representation of the molecules is the same length, the content can change, right? Because it will depend on how much molecule you have in there, how diverse the data set is, and so on and so forth. So I'm not going to get deeper when it comes to learned features. But we will focus on the classical machine learning. You can also combine both. So for the sake of expressivity, especially if you have a good estimation of what properties can be very important for your specific needs, for your specific target or endpoint, you can add or combine learn and fix properties together for better performance of your model. Now, the classical operations in predictive modeling include, the data collection. Obviously, you need to get the data from somewhere. This is a very critical part. Sometimes we are so excited about creating a model that we forget about the quality of the data. The data quality is very important. We usually say garbage in, garbage out. If you put garbage data in, that is not standardized, that has a lot of wrong values. You have duplicates, with different measurements. I've seen cases where, usually when you are doing you're testing your molecules, you want to try maybe twice or 3 times for the same molecule. You might select a few molecules, when you have, a million of them or, 10,000 of them. You're not going to replicate all of them necessarily, but you might want to try a significant number that you replicate, right? So that allows you to then measure the standard deviation and also understand how reliable your experimental setup is. So, and I've seen cases where because of the experimental setup, because it was not very appropriate, you will have a molecule, for example, that an assay that measures the degradability of a molecule in soil, for instance, right? And then one of the measurements for the same molecule will say the molecule is degrading completely, and the other measurement will say the molecule is not degrading at all. So that's a significant problem. So you want to remove these examples, right? But you also want to question the reproducibility of your analysis. Is very of your experiment. So if the experiment is not viable, if it's not reproducible, you cannot expect the predictive model to do any kind of magic here. So that's why we say garbage in, garbage out, right? That is very important. As a matter of fact, the overwhelming majority of the time spent in predictive modeling is spent in data collection and preparation. So once you've done that, you split the data. Usually you want to train the data, the model on a specific set, and then test it on a set of molecules that were not used for training, right? So you want to make sure that it can generalize, because the idea for you is that you're going to train it on some molecules so that you can test it on other molecules for which you don't even have the results yet. So to be even more precise, or to increase the chance of having a better model, right? A well-performing model. Instead of splitting into test, we will split into train, validation, and tests, right? So during the training, you will train on the model on the training data, you will validate it, and that's usually helpful when you are doing hyperparameter optimization or when you're training several models, right? Training over time. And then at the end, after you get the best of the models, then you can test it, right, on the test set. So then you select the model and train. You can select, different type of algorithms. You can go with deep learning models. You can go with, classical machine learning models. You can even try, make all of them together. Not every single architectures, but various architectures, deep learned or classical, right? And once you do that, and you find the best model, then you have to validate it. Again, you validate it on the test set. don't be confused here when I mentioned validation here and validation here. Here, this validation set is during the training, and the test set is where you actually validate your molecule on external data. You need to understand what the metrics are, select the appropriate metric, and that's what is going to be used for you to even compare models, right? So then in terms of the application, you predict the biological activity on new molecules, and then you interpret the model if you want. It's usually a good idea to be able to interpret the model. So that will allow you to detect maybe the biases in the prediction. It will help you also, understand what features are contributing to, activity in general, for the whole set of molecules, or maybe for a specific molecule. Then you want to validate it again on some other data that, was never seen before. So here you're working on data that you maybe gathered, over the last 5 years or whatever. Now, maybe in 10 days, in a month, you have a new batch of data that is coming in. It might be a set of molecules that is not really similar or close enough to the molecules that you use to train. So that means they are not necessarily in the applicability domain of the molecule of the model. So you want to make sure that you can test those and see if the model is still performing. If the model is not performing, that means that you probably want to retrain the model to expand its applicability domain to this new series of molecules, right? So that's where we are. And I'm going to stop here and take any questions before I go to the practical part. Is there any question? I hope that this will be kind of interactive. So please let me know if you have any question. If not, we're going to move along. Okay, so the next thing that I'm going to do is I am going to share what we call a notebook. We will go through this together. It's not going to be exactly live in the sense that you're not going to be running the code in parallel, but as I mentioned, I will share the whole data so that you can replicate the operation, right? So what we're going to do here is basically train classification models that can predict a compound or molecule's ability to inhibit the Plasmodium falciparum's growth, and that will be the DD2 strain. So with respect to the minimal number of molecules, that's a great question, by the way. It's important to have as many molecules as possible. It's important also for the data set to be diverse. So you don't want all the molecules to be from the same series. You don't want all the molecules to differ just by a single, functional group or single atom. So you want them to be diverse, as diverse as possible. I will get into it a bit later. The minimum recommended number of molecules, it really depends. If you want a molecule, a model that is very local. So imagine you have found a scaffold, right? That is very active. You have a set of molecules that are from the same scaffold, and you want to generate additional molecules from the same scaffold, right? You might get away with just having a few hundreds of these molecules and building a local model. If you want to build a global model that is applicable to a large to really diverse set, you might go you might need a few thousand, if not more. Now it will also depend on how complex the endpoint is to model. Certain models are quite easy because of the simple relationship between a mathematical relationship between the properties of a structure and the endpoint, right? But there are others that are very complex, and for those you will need a diverse set. You will need to try, different methods of representation. You might need to combine, fingerprints with, molecular properties and so on and so forth. So, but we will get into that. So I am using Python here, right, as a programming language, and the idea here is that we will use Python and associated libraries in order to generate, the model and to evaluate. Right? So, sorry, I have to accept a few people here before we continue. All right. So I'm using VS Code. VS Code is an IDE that is very good for just text, but also for programming, right? For writing code and testing, and it has an environment that is really really neat, right? So you also have the terminal for those who are familiar with Linux. You have the terminal where you can really do a lot of work. But I'm also using Jupyter Notebook. Jupyter Notebook is a framework that allows you for easy interactive programming, right? So instead of writing the code into a file that is difficult to read and whatnot, so I'll write the code in the Jupyter Notebook. But I have in this folder that I will share with you, I have modules that are written that can call some of the functions that implement some of the functions that I'll be calling in the notebook. I will also share a list of dependencies that you need to install before you can run this code. So with respect to data preparation, first of all, I import the necessary modules here for processing. I've split the project into two different parts: the data processing and the training. The training notebook will also contain evaluation, explanation, so on and so forth. So I'm loading all the different packages that I need. Pandas is for tabular, so representation of tabular data. NumPy is for manipulation of vectors. Matrices is for basically linear algebra. RDKit is the package the Cheminformatics package that I'm using to draw molecules, but also calculate properties, convert smiles to different formats. And I'm also using Maldred. Maldred is another package that implements several physical chemical descriptors. So I'm setting the seed here. This basically is a number that kind of control the operations, right? The way, so especially when you are doing operations that can randomize, that can use random sets, maybe to select or to mutate some object, right? So for the sake of reproducibility, you want to set the seed so that others can replicate the results, right? So next, I use Panda to load the data. So the data looks like this, right? The data is from a paper that was published earlier. Basically, they gathered the PubChem bioassay with ID 2302 that was
Yannick, excuse, we can't see your screen.
You can't see my screen,
please? Yeah.
Oh, that is strange.
You are stuck in ask a question slide.
Oh, my bad. Yeah, okay, let me do that. Yeah, because I changed. Okay, I'm gonna share this entire screen. I think I was sharing just a tab. My bad. So, okay, so here we are. So just to summarize, let me know when you see my screen, okay?
Yeah, it's okay now.
All right. Thank you. Thank you so much. Thank you. So so you're seeing it properly. Okay. So here is where I'm loading some of the libraries that will be used for my programming. I'm setting the directory, where the code and the other folders are saved. I am importing AutoKit. I was mentioning AutoKit earlier, which is used for molecular operations. Basically, it's one of the most popular cheminformatics libraries to date. And here's why I'm setting the seed, right, to be able to reproduce operations. Here, I'm using Panda to read the tabular data. And these are the smart strings, right? You have the PubChem IDs, you have the primary outcome, and then you have the percentage of inhibition. Basically, the percentage of inhibition is what has been used to determine whether a molecule is active or inactive. Generally, here, if the percentage of inhibition is above 50, or is equal to 50 or greater, then it's active. If not, then it's inactive. So when you're doing the cleaning, it is very important to, first of all, remove all of the samples, and the sample is identified here as a row, right? All of the samples that don't have a SMILE string. If you don't have a SMILE string, you don't know what the molecule is, right? So you need the SMILE. Yes.
Yeah. Still, from my side, I cannot see your screen. I don't know if other members they can see.
We can see Lucas.
Yeah.
Oh, okay.
Yeah, and maybe you want to adjust your Chrome, or you want to log off and join again. I'm not sure what, but does anybody else have an issue seeing the? No. Okay. So we remove the empty the line with the empty smiles, right? So, and to see how that is done, basically you can look at the clean data function in the preprocessing module. The preprocessing module is here. And again, I'm going to share the document. So I will provide my email address, and feel free to contact me at that address, and I will share the URL with all the the clean version of the code. So if you look into the preprocess modules, because we in Python we basically create modules that encapsulate features and functions. It makes it easier when you're running your code. It's make it easier to debug, and you also don't want to have a gigantic file with 2,000 different functions in them. So you can group them by theme. I have one for preprocessing, I have one for splitting, I have one for visualization, and so on and so forth. So basically, what this thing does, this function, it takes in the data frame, the target column. The target column for our classification will be called DDS activity CLS, DD2 activity CLS, right? For DD2 activity classification. Now, what is going to happen with the clean here, since I have active and inactive, I will want to turn this into ones and zeros. One will be for active, and zero will mean inactive, right? So since we are not doing any regression analysis, we are actually just predicting whether it binds or not. We are going to remove this one. We're going to clean, obviously, the SMILE string. So, and sometimes when the chemists are, a lot of you here are chemists, so you will run your test. You will, usually the molecules or the substances will be salts. For example, you have sometimes a molecule or the substance is is maybe chlorinated or sulfated or whatever. So they will often report molecules that you see here. This molecule here, when you see the dot, it means it's a salt. So it's a chlorinated molecule. So you want to remove the chlorine, because at this point we assume that the activity is linked to the main molecule and not the chlorinated part. So what I'm going to do is I'm going to fragment the molecule, the substances here, provided by the SMILE string, and just get the largest fragment for each molecule. Okay. the largest fragment here. So, and then I'm sanitizing it, meaning I'm going to clean the SMILE string. So this SMILE string here shows a representation. So you have a carbon that is, in a ring. That's why you see the number here. And then it's, linked to another carbon using a double bond. The double bond signifies that it's the equal sign signifies that there is a double bond, and then it's linked to another carbon atom, and so on and so forth. So I'm going to clean it, put it in a way that is easy to manipulate later. So with respect to the labels, we have to check the integrity. So sometimes you will see duplicate. So you see some molecules you see some lines where you have the same substance or the same molecule. Sometimes you may even see the same molecule basically, but it's different SMILE strings. So different SMILE strings, but it's the same molecule, and it's different. They all have a different CIDs, right? So you want to make sure that you take one molecule and only one molecule, especially in classical machine learning, one molecule per, or one SMILE representation per molecule, right? So, and sometimes you would have replicates, as I mentioned earlier, and the replicates might indicate different outcomes. Imagine you have the same molecule. Imagine, just for the sake of the discussion, that these two molecules were the same, right? And then you have one measurement says it's inactive and another says it's active. What do you do? If you only have two, imagine here, since we are using the percentage of inhibition, right, to classify it as inactive or active. If, the percentage of inhibition for the first molecule said 4, and for the duplicate it says 80. Those are at, the valid values, the range is between 0 and 100. You cannot have 4 and 80. So that's something went wrong. So you want to remove those, right? If one was 49 and the other was 67, usually you want to do an average, and it's going to be a little bit over 50. So that means you can see classified as active, and then collapse those two rows into one. Now, to be more strict, you might want to, collect or group the data frame once you have cleaned the SMILE strings and created what we call the INCHI key. The INCHI key is a unique identifier, right? So this is the INCHI key of the largest fragment. Once you have all of those together, what you want to do is then group the whole data set by INCHI keys. So for some INCHI keys, you're going to get only one. The group is going to only have one instance. In some cases, you might have 3, 4, 5 instances, meaning that for the same compound, there were 3, 4, 5 measurements, right? And then you want to see what are the values, right? So you might identify outliers and you want to get them out. And if you remain with, let's say, 2 out of 5 molecules or 3 out of 5 molecules that have a range that have values around the mean, right? Not too far from the mean of the whole data set, then you can just, do the average and then use that as your value before then you assign whether the molecule is active or not. So I'm not going to go any further, but here it prints the number of rows with undefined label after we've cleaned the data is 0, right? So the number of molecules with missing SMILEs was 13. So we have removed 13 out of 13,533. That's why you will see later on that we have 13,520 because we removed those. Now, the number of unique INCHI keys, we have 13,520 molecule rows with valid SMILEs, but we only have 13,450 unique INCHI keys. That means, a little bit less than, a little bit over 50 or so, 70 or so are duplicates, right? So we're going to remove them. I haven't applied the strictest duplication removal here, but yeah, so that's what we did here. Now you have the number of duplicates that were removed. You have an example here of a duplicate, right? So these are the indices or the compound IDs actually the indices of some of the duplicates. You see, the SMILE strings here might have been different actually, but then the INCHI key is kind of a barcode. It provides a unique identifier to molecules. So you can have SMILE strings that are different. That's the disadvantage, but also the advantage with SMILE strings. You can have SMILE strings that are different. Look at them as synonyms, right? But they represent the same molecule. So that's why you want to compare the INCHI keys here when you are looking for duplicates. So you have those two duplicates. Luckily, you know, they are all classified as active. So we are only going to take one measurement. And here, same thing, those two duplicates, they are all classified as active. So we're going to take, you know, just one of them. So now, with respect to molecular representation, once we have our data set, so what is going to be interesting to us is going to be from now on the SMILE string, here, the largest fragment of the SMILEs. I added hydrogens here. This molecule that we're going to use, right? The R-mol is the whole molecule without fragmentation. And here is the molecule object implemented in RDKit, right? That is going to represent the largest fragment. So that's what we're going to use from now on. And the SMILE string, as well as the INCHI key of the largest fragment. Now, with respect to the representation, I mentioned earlier that we have, you know, you can have fixed representations and learned representations. And here we're going to focus on fixed representations. So you have the constitutional descriptors, basically such as, you know, the molecular weight, the number of atoms, and you also have the geometrical descriptors. They basically are related to the molecular shape, size, and symmetry, right? So they describe it in various forms. You have the topological descriptors that are based on the connectivity of the atoms, right? Different indices. So they look at each atom and see how connected it is to others, and so on and so forth. So you have the electronic descriptors that are related to, you know, the density, the energy level, and whatnot. And then you have the constitutional or functional groups. So here, for example, you might be looking at the number of amines, as I mentioned earlier, the number of carboxyl group, the number of, the number of nitro groups, and so on and so forth. So I have implemented a featurizer here in this module called featurizer. Basically, what it does is it takes a molecule and, you know, based on user-specified list of features, it will calculate all of those features and put them together for all of the molecules. So, for example, here I calculate the molecular weight, the number of aromatic rings, and so on and so forth. And a lot of these indices that have to do with the topology. I'm using Modred and RDKit for that. But also with RDKit, I'm calculating the number of occurrences of certain functional groups within each molecule, right? So it takes about 2 minutes to calculate over 200 features. Now, one interesting thing is sometimes you might have elemental features that don't mean that much, right? But by combining them, you know, you might have a huge signal here that is going to be, you know, helpful for modeling. And you can customize your features, right? In this case, for example, I added the number of halogen atoms and the weight ratio, right? In some cases, you know, it can be very important because halogen can impact the atomic properties of a molecule, right? So, you know, depending on the scenario, in agrochemical design, for example, we'll say, okay, we want the ratio of the weight, the total weight of halogen atoms to be no more than 30% of the total weight of the molecule, for instance, yeah, for a number of reasons. The fraction of sp3 carbon is also very important. So sp3 carbons tend to be more hydrophobic. So when I say that, you know, you understand that they can, you know, impact the protein binding and so on and so forth. I also added, you know, a value, a Boolean value that tells if a compound matches the Lipinski rule of 5 or not. So here is an example for the first 2 molecules of the data set. These are the values for the you know, the features that I added, right? Now, here I'm cleaning. So here, what I was doing was computing the properties, right? Here I was cleaning the data set before having all of this, the features. So I was just removing the bad smiles strings and removing the rows with empty smiles. Here I'm actually cleaning the data set. So I'm going to remove the values that are nan. I'm not going to standardize at this point, but I'm going to remove the values that are nil and do some additional work here. So you can look at the code in the pre-processing. And also, one thing that is very important, because we want to use as little resources as possible, right? So the data frame here was originally used with a floating number. For those who know a little bit about the arithmetic here, so it was using floating number with 64 bits. Usually those are helpful when you are expecting really large values in your data set. Here we're dealing with small values that can be represented with a smaller flotation number. So by that, I was able to decrease the memory usage. By reducing the memory usage here, I was able to gain about 74.6%. 74.6% in terms of memory. So I have more memory now to use for subsequent data compared to before doing before calling this function here. Now, let's go to the fun part. Often you want to see what of your descriptors are really correlated with your target. So that might give you an idea of what features are important. But it's important also to note here that just because a single feature doesn't have a high correlation with the target, it doesn't mean that it is not important. Sometimes that feature might be important when you collapse it with other features. So the molecular weight might not be very important, for example, for a specific outcome, but when you combine the molecular weight with the number of bonds and number of aromatic atoms, number of, maybe chlorine atoms, maybe all of those together are going to form a very strong feature, right? So that's for the model to decide. So, but it's already really interesting to see that, for example, this topological index here is highly correlated. Obviously, the number of aromatic bonds is also highly correlated. So the higher and here I'm measuring the absolute correlation. So, so it could be a positive correlation or a negative correlation, but here, just for the sake of comparison, I'm just looking at which ones are highly correlated to the target value, right? So the higher this value, the more correlated the feature is to the target value, right? So, and another thing that I'm doing here is I'm plotting the correlation numbers for the added those 4 or 5 added features, the custom features that I added before. So you have them here. Of those, the feature that correlates more is a fraction of sp3 carbons, right? Okay. Now, with respect to the visualization, the Lipinski rule of 5 is a set of a few rules that were determined, I think, late in the 90s, for that says that molecules that fit or that matches those constraints are, very prone or very likely to be drug-like. So with a radar plot, especially when you're comparing active versus inactive, it gives you an idea of the distributions, right? Of the different values. So it lets you know how far or what is the range of the values, right? So this basically, the area here is basically the area that you're targeting, and sometimes the values might go a little bit above, right? So another way of visualizing the distribution of the functions of the descriptors is through box plots. Basically, the box plot can give you an idea, for example, of what molecules, what features, for example, are differently distributed between the active and inactive class. In this case, for example, when you look at the number of aromatic rings, remember earlier, it was a feature that had a high correlation, right, with the target value. You will see here that it occupies a fairly, so 50% of the values are within this box here in active molecules, right? So, but for inactive molecules, the values occupy a wider range. So that is an indication that the distribution is a little bit wider here. And it's an indication that it might be an important feature. In some cases, the distribution are the same. Like here, for example, number of hydrogen bond donor, the distribution are almost the same. So basically, you have this endpoint. These circles, dictate or specify what the outliers are. But then you have generally the lower value, the higher, the max value outside of the outliers. And then this tells you, the median line here tells you that 50% of the molecules have a value below this value here, this threshold here, and 50% have a molecule above. Now, in this case, also the features occupy basically are distributed within the same interval. However, because the median line is lower, it means most of the molecules, right? Most of the molecules are pushed towards this higher part. And here, most of the molecules are pushed towards the lower part, right? So I have within the notebook a lot of text to explain what I'm saying here. So if you don't understand one of the things that I'm saying, you can ask questions, but you can also I put quite a bit of text here to let you to help you understand the concept here. So with respect to the functional group, you might also want to know which functional groups are very important, right? So you can do a plot. So here, what I did was I looked at the occurrence, the number of occurrences of each of these groups. So piperidine, the sulfonamide, alkylic carbamate in each of the molecules, right? And then I count. So for the molecules that are active, how many molecules, how many times do I see, let's say, a pyrrole? How many times do I see a sulfonamide? And for the ones who are inactive, how many times do I see a sulfonamide? How many times do I see a pyrrole? And we get a plot like this, which basically tells us in terms of and I've normalized the numbers. Instead of plotting the absolute numbers, I've normalized the numbers so that we can, just have them between 0 and 1. So you see the carboxylic group relatively, it occurs more often in inactive molecules than active molecules, right? So often you want to see a big difference between those two lines here, those two bars, and that will be probably an indication that a certain functional group is important, right? In this case, the sulfonamide group is one of the important ones. Now, I talked about scaffold earlier. Basically, a scaffold gives you it's a method used in cheminformatics to group molecules together. So it basically is just a representation of the circle, the rings within the molecule and the linkers. We don't talk about the side atoms, right? I will show you some examples, and that helps you put molecules together. It could happen that molecules that have the same ring, same ring system exhibit similar activity. Maybe they are all active, right? Maybe they're not. So that's what you want to know. So usually what you do is you can break molecules within scaffold to see also what's the distribution of your data set. Is your data set diverse enough? If the molecules are the same scaffold, you will only have one bar here. But if they have, if it's diverse enough, you will generally see several scaffolds here. So basically, I, computed the scaffold, the Bemis-Merkel scaffold for all the molecules. And then I grouped them together. I computed the size. So basically here you have about, so you have to count the number of molecules that, have a specific scaffold. And there is one scaffold that, there's one scaffold group that has about 5,000 molecules. And you have, because the data set is so diverse, you have 6,958 different scaffolds. That's huge. That speaks to the diversity of the of the data set, right? So usually you want a high number here. Could be problematic sometimes. Sometimes it could actually be helpful because it will help you develop a bigger, maybe a more powerful method. So these are the scaffolds here. So you see here, there are no side atoms, no side chain atoms here, just the rings and the linkers. And then you can actually go a step further. You can decide that you don't even want to take into consideration whether a ring is aromatic or not, or whether you have heteroatoms. You can actually decide to make it so that the scaffold only have rings, carbocycles, and carbon atoms. So you replace the N by carbon, N by carbon, N by carbon, and instead of quinoline, this is going to be a set of two cyclohexanes linked together, a few cyclohexanes, right? So this scaffold group has 161 compounds. So there are 161 compounds that contain this scaffold here, right? In contrast, there are only 41 that contain this scaffold. Another method to analyze or to visualize the data is by clustering. So when you cluster, you don't necessarily look at one specific scaffold. You just look at compounds that are similar, right? And there are many metrics, many ways of calculating the similarity. Here we use the Tanimoto coefficient, and we use the fingerprint. So we're using the Morgan fingerprint to basically take a molecule and turn it into a vector of length 1024 bits. And each of these bits tells you how many times a certain feature occurs in the molecule, right? So, and then you use the Tanimoto coefficient to kind of see which molecules are similar. So the molecules that are similar for each cluster has a centroid molecule. And then once you pick the centroid molecule, then you go and look at the rest of the data set and you find what molecules are similar enough. You can specify a cutoff. You can say, I want a cutoff of 0.3. So every time I get a centroid molecule, which is the head of the cluster, every molecule that has at least a similarity of 0.3, which is very low, will be part of this. So in this case, generally, you will have a small number of clusters, but you have clusters that are very large. Here, the cluster, for example, the largest cluster has almost 5,000 compounds in it, right? And there are 513 clusters. You do the same thing and you vary the cutoff. And here you have, because I increase the cutoff to 0.4, that means it's more restrictive. So the cluster will tend to be smaller, and you have more clusters. Now you have almost more than triple the number of clusters. Now, if you increase the cutoff too much, then you end up having too many clusters, right? That might be a problem. Sometimes it's good when you have molecules that look alike, even if they are not too similar, because there might be some properties that are not maybe similar to the scaffold. There might be some small properties or small nuances, small similarities that are difficult to visualize, but that actually maybe transmit a certain signal. Maybe if a carbon if an hydroxyl group is adjacent to a carboxyl group, then it will signify or will increase the chance of the molecule to be active, for instance, right? If you're just looking at a scaffold or you're looking at molecules that are very similar, you might not see that. So now I'm going to show you 10 molecules of the largest cluster. When I take the similarity cutoff of 0.3, you will see that here they're still kind of different. They are not dissimilar, but not as similar as you might want. So they don't have the same scaffold, because here I don't regroup molecules based on scaffold, but just based on similarity. So here are some examples. But then when I increase the similarity cutoff to 0.7, you will see that the molecules are more similar here compared to what we have here. So there's a trade-off, right? You want to have clusters that are large enough. You don't want to have too many singletons, right? There are advantages and disadvantages of as you are increasing the similarity cutoff here, and those are described here. Now, To visualize the data set, you can also do what we call a UMAP projection. Now, I was mentioning earlier, for the Morgan fingerprint, you have over 1,000 bits. If you want to visualize the molecule in this, in the real world, you would have to use, you have to visualize the molecule in, 1,024 dimensional space, which is impossible. We cannot do that. So what the UMAP projection does is it takes such a large vector and it compacts it. It does what we call dimensionality reduction. It compacts it into a vector, into representation of a 2D representation or 3D representation. In this case, I was shooting for the 2D representation, and it tries its best to mimic or respect the actual similarity or distance, if you will, between two molecules. So the similarity between two molecules in the 1,024 dimension will be almost the same in a two-dimensional space here. So that's the goal. So you will see here that the active and inactive are not, it's not easy to differentiate them, which might be a problem, or might make the process more difficult. So I'm plotting different styles here. Here I'm using a different type of fingerprints, which is much smaller. It only has 167 keys, 68 keys. And with that, I get a different plots. Here you will see that they are more active compared to more active molecules compared to inactive. Here you have some clusters of active molecules, but for the most part, active and inactive occupy a fairly similar space. Other methods to assess the diversity of the set are, similarity metrics. You have the Euclidean distance, the Manhattan distance, the cosine dissimilarity, and so on and so forth. So now we come to the data splitting, right? Here I'm using cross-validation. We are going to use cross-validation for training the data set. We're going to use cross-validation, and this usually is a better technique because it allows you to train the model several times over different data sets, and it makes the model more robust. So that's the first part, right? The second part, which should take less time to explain, I guess. We have only 15 minutes. Is about the training and invalidation. So here I'm gathering all the data. So I generated molecular features using Mordred and RDKit. I also have fingerprints representation with Max and Morgan, and I have my splits here, right? So what I'm gonna do is build models. But before that, there's something that I want to mention here. When you're doing your splitting, it's usually very important that the ratio of active against inactive is consistent, right? You're going to try to do that as best as possible. Usually, you have packages that allows you to methods that will just look at the number of actives and inactive, and every time they're going to do the splits, they are going to make sure that the ratio of active versus inactive is the same for each split, right? And but it's difficult when you take into consideration the molecular features. So fortunately, we were able to mimic the same distribution. So they are more active than inactive. Usually it's the other way around, but here they are about 1.4 times more active than inactive, right, in the whole data set. Now, in each of the folds here, or the splits, right, the ratio is almost the same. It's conserved. It's conserved, right, with a few exceptions like here, right, and here. But other than that, it's fairly conserved, which is good. So in order to train the model, we are going to use a light GBM. So it's a gradient boosting model implementation. And I provided the link so that you can see what they are, and even have some tutorials here on how to implement them, right? So, and we are going to use a hyperparameter optimization, which is a technique that looks for the best parameters. So look at the parameters are as the tools that will dictate how good a model is. It's like when you're cooking, right? You know, you will have your salt, you will have your pepper, you will have, you know, your condiment. Now, but you want to actually know what is the quantity that you want to put for each, you know, ingredient to make the perfect sauce. So basically, that's what we're doing here. And instead of going and, you know, doing what we call a grid search, so you take all the possible quantities, and then you permutate them to get a bigger set of combinations. We actually use what we call Bayesian optimization here to basically more intelligently and you know, more intelligently and strategically find the parameter combinations that are suitable. So with the light BGM model, I use the molecular descriptors, and I trained them and I validated, and I got the best parameters. So you can usually get the best parameters, meaning the parameters that leads to the best models. And then you have the validation score that is 0.4. And I'm using the balanced accuracy score here, which it will be defined a bit, you know, later, right? So now, once I have that, I want to retrain the model on the whole train and validation set, because I was training on the train set, validating on the validation set. So I want to combine the two and then train the model there and then use that model for testing. Here, I'm talking about a few of the metrics that are important to compare the models. So you have the precision and the precision and the recall, as well as the file out rate, right? The precision will tell you of all the prediction that you made, how many were made correctly, right? How many of the positive samples were the samples that you predicted to be positive or active are actually active. And here, for the recall, it will look at all the active molecules and figure out what is the percentage of those active molecules that you actually predicted correctly. And we can use those to test how good a model is. We have here what we call the confusion matrix. And the confusion matrix basically will give you an idea of how well the model did. So on the x-axis, we have the predicted label, the label that were predicted by your models. The true labels are on the y-axis. So basically, this says among the inactive molecules, right, in the test set, right, and there were about, I would say, 558 that were inactive. You predicted 219 correctly. So inactive. The inactive molecules that were predicted as inactive. So basically less than half. Now, and then you predicted 124 incorrect. You predicted 239 incorrectly. Among the active, and there are about 794, 670 were predicted correctly, and 124 were predicted incorrectly, right? So, and you use that to calculate the precision score, the recall, and so on and so forth. So sometimes, depending on what you need to achieve, you might want to prioritize the false positive rate, right, over the recall, right? So maybe you have a situation where you have limited resources for testing, because usually when you predict these molecules, when you predict these outcomes, you take the molecules that are active and you go and test them again to confirm that they're active, right? You test them now experimentally. You might not have enough resources, right? What you want is maybe a model that, yes, might not find out all the active molecules, but you want to be confident that the ones that are predicted active are truly active. In that case, you will want to have a higher precision model, a model with a higher precision and lower recall. In case you have enough resources and you have other predictive tools to quickly sort out molecules, right? You want to get as many active molecules as possible. You want to identify as many active molecules as possible from your input data set. In that case, you might want a higher recall. Usually there is a trade-off. If the recall is higher, the precision is going to be lower, and vice versa. So in order to really understand what the model does, right, overall, and also as you're varying the threshold, you can plot the ROC, the rock curve, basically, which gives you an idea of how good the model is, irrespective of the threshold that you select, right? In our case here, what the author did is they took a cutoff. So, and in our train, so they said 50% or more means that the model, the molecule is active, and 50, lower than 50% means that it's inactive. So in our case here, we predict the probabilities, and we say if the probability of class 1, or the probability, is over 0.5, and remember, it varies between 0 and 1. If it's over 0.5, it means the molecule is active. If it's less, it means the molecule is inactive. Now, in some cases, you might want to move that threshold. Maybe your model becomes better when you reduce the threshold, right? So in that case, what you want to do here, you will see you vary the threshold, and then you will see that your precision in this case, because we have more active, the precision is higher when you increase the probability threshold. So increase the probability threshold means you are very selective. The compound has to have a strong signal, a strong probability of being active for you to select it. So that means when the model is predicting a molecule as active, the likelihood for it to be active is very high. However, it also means that you will lose a lot of the active molecules. You won't see them. And you will see here, as you are increasing, the precision is increasing, the recall is decreasing. Right? So if you have only limited resources and you don't want to, you don't want to have a lot of false positive, meaning molecules that are predicted as positive but are not, you want to go with a higher precision, but then you will lose in terms of the recall. So we plot these values. Generally, this is the perfect model that predicts everything correctly for the rock AUC. The rock curve will be this black square here. So the closer the summit here of the blue line is to this point here, the better it is. And the AUC curve, the AUC, or the area under the curve, measures the space here. So it's about 0.61 and 681, which is not bad for a model. And I would like to specify here that just for the sake of training, I used 25 steps for training the molecule, because you do that iteratively, right? You do that iteratively when you are doing hyperparameter tuning. Generally, you want to go way past that. You might want to do 100 or 150. It depends. It doesn't guarantee that the model is you are going to find a much better model, but usually the chances is higher. The chances are higher for you to find a better model. And this is the prediction recall curve. So basically, you're varying the threshold, right? As I mentioned earlier, the cutoff or the probability threshold, and you want your model to be somewhere here. So the top part of the model, so the middle of the curve, you want it to be here. So, but for this model, it's kind of below. So when you measure the curve or the area below this line, below this curve here, you get 0.724, which is not bad at all. Now, in terms of feature importance, with the light GBM model, you can see what features are more important, right? So, and for this particular type of architecture, for the gradient boosting machine, you can detect the feature importance either by how many what properties is used to split the trees that you are using to build your model, because this is a tree-based algorithm. For those who don't understand, there are links here to describe what the models are, or you can decide that you can select or calculate the feature importance based on the gain. So how much gain do you get? How much does the model get by adding this feature here? So this AETA has the most important feature. So if you remove that from your descriptors list, the model is going to lose its performance quite a bit. And so there are the mod log P, basically the octanol water partition, which deal with the absorption, dictate the absorption of the molecule is a very important one. So these are a bunch of topological features that are not easy to understand. So I'm just going to focus on the mod log P here, and you can see that it's also here, right? So I tried different models using here the max fingerprint, and this is the normalized confusion matrix. I use the ECF, so the Morgan, and I also got I got a number. So here in this case, I combine the descriptors, the Morgan fingerprint, and the max fingerprint. So I have a larger dimension, right? So, and here I got a model that was slightly better. It had a better recall, but not necessarily the best precision score. So you can then compare the ROC curves here for all the models. So it appears that the one with the descriptor is not too bad. It still have the highest area under the curve. So the blue one here, and when it comes to the precision recall curve, the best model is still the one with descriptors. Again, if you train longer, things might change a little bit here. But you could see here also that having just the descriptors that are, they seem to be to express a lot more, to be more informative than just having a bunch of ones and zeros, right? Or integer values for Morgan here. And combining a lot of those to build one model is not necessarily the best solution. And here I'm talking about the explanation. You want to make sure that you can explain your model, you can interpret it, right? Yes, I will share it. I realize we're already at time, but I will definitely share it. I will provide my email address here. Basically, please send me an email, because I couldn't get all your emails, right? Send me an email to this address and I will share you the link to the code, please. I'll definitely do that. It's very important that I share this with you. So here I'm explaining basically in different ways how the different features contribute to the prediction. So here, the one in red push the prediction, or if you want, the probability towards the positive, and the blue ones push towards the negative, right? So basically it means that the mod log P is contributing quite a lot